I'm going to finish off topic 3 here. We spent a lot of time talking about different types of microscopy. So light microscopy, fluorescent microscopy, electron microscopy, probe microscopy. I kind of have a few things just to really talk about to finish off this particular unit. And these are not related to microscopy. They're related to a few other things such as cell culture. So first thing to consider is, why do we want to grow cells in the lab? So there's actually quite a few reasons besides just studying them and doing research on them. We sometimes have to grow things for diagnosing diseases. So for example, a few years ago I had strep throat, and they take a little swab of your throat and they send it to the lab, they grow it up, and they diagnose it as strep throat. Sometimes we produce vaccines, growing cells, and we have many, many products that we like to grow. For example, if you make yogurt at home, that's basically a cell culture. If you make wine or beer at home, that's a yeast culture. So there's lots of reasons why we grow cells in the lab besides just studying them, doing them for school. So how to grow these things? Generally, we need to feed them, and they have physical requirements. That includes food and temperature and things like that. Or sorry, physical requirements, temperature, pH, osmotic pressure. Food part is the chemical requirements. So I'll come back to that in a moment. So temperature. When we grow things in the lab, most of our organisms grow at about 30 to 40 degrees Celsius. And that's because, well, human body temperature is 37 degrees. Most organisms are most comfortable in that particular temperature range. Once you start getting around 50 degrees Celsius, most organisms, the proteins will actually melt and denature, and it makes life not possible. There are some organisms that grow at much higher temperatures. Certain types of bacteria, certain types of archaea, but most organisms, 50 degrees is about the maximum temperature for most organisms. Optimal growth about 37, minimal growth about 4 degrees. Again, there's always exceptions. There are a few things that will grow below freezing, but generally once you hit freezing, water can't move around, so things can't really grow. So a word that I want you to know is this one here, mesophile. And that includes most organisms fit into that particular category. Meso means something like medium. So medium or average type temperatures. Another term to know is a thermophile. These are organisms such as certain bacteria and archaea that might be able to grow at high temperatures. Those other terms I don't want you to know about. Or well, you can know them, but you don't need to learn them. Let me show you some temperatures. So for things that we're growing in the lab, we're going to grow E. coli in the lab, and E. coli's optimal temperature is actually 40 degrees Celsius. So you're probably thinking, why not 37? Well, not everything is human. E. coli actually grows in birds as well, and birds tend to have hotter core body temperatures. You can see I have a couple of pathogens listed there. I have Mycobacterium tuberculosis. This is a human pathogen. It grows in the lungs, causes tuberculosis, and its optimal temperature is 37 degrees. There's another one here I have listed, Listeria. I think I have another slide on that. Let me just take a look. You can see its optimal temperature is 30 degrees Celsius. Why do I have Listeria listed? Because you may have heard about it in the news. Quite often, Listeria will have these recalls. Every year there's a Listeria recall, one or two across Canada, some sort of contaminated lunch meat, or I found this one here just by a quick Google search. This was some sort of macaroni salad you can buy. I'm not familiar with this product, but it was recalled a few years ago. And the thing about Listeria is that most organisms grow well at that 30 to 40 range. Listeria most happy at 30 degrees Celsius, but also grows very well at fridge temperature, so about 4 or 5 degrees Celsius. And so it's a serious concern with the food industry because it grows really well at fridge temperatures. So besides physical requirements, we also have chemical requirements. This really just means what food it requires, food, nutrients, vitamins, things like that. One of the requirements that that is essential for many organisms, of course, is oxygen. And we use the word aerobic, meaning organisms that require oxygen. And I'm showing you this little slide here. I couldn't find a great picture of it. So this little cartoon was a little bit better. And it's showing these organisms growing in these special tubes. And in these special tubes, the top part of the tube has oxygen, and the bottom part of the tube is anaerobic. So you can see we're ending up with quite a number of different types of organisms. Some of them are obligate aerobes. So that means they require oxygen. Obligate means obligated. Some are obligate anaerobes. That means that they require no oxygen. Oxygen will actually kill them. And then there's other things in between. Another one is facultative anaerobes, which means they're okay if there's oxygen, there's okay if there's not. And E. coli would be included in that particular category. So we do have one bacterial organism that we're going to grow in the lab that is anaerobic. And I just want to point you to this thing on the right here first. This is called an anaerobic glove box. You may have seen something like this in movies. Sometimes people are handling biohazardous waste this way too. The researcher is separated from the samples, but you can actually get these anaerobic glove boxes, which are a giant box with, it's usually flooded with nitrogen or something like that, and then the samples can be handled with those gloves. Those are expensive. So what we actually use is this thing here called a gas pack system. So this gas pack system you can see on the left is kind of like a big jar, and it has a seal on the top. You can see it's clamped and it's airtight. And what we have right here are these things that are called gas packs. You can buy them, and they will chemically react with the oxygen and basically suck it out of the system there. So we're going to grow a organism called Rhabdosporillum using the gas pack system, and I'll take a little video of it for you so that you can see it for our lab. That would be lab 4. So you're probably going to see in the lab I'll be handling some E. coli and some other bacterial organisms, usually in test tubes. This is a pretty common way to grow organisms, either bacteria or cells. And you can see in this case here we've got a few different species. This one here is clearly E. coli. And you can see there's a recipe there, something called terrific broth. I kind of like the name balance, so I wanted to show that to you. You can see the terrific broth, there's a whole bunch of things in there. The big thing that's important is this yeast extract. So what does that mean? It means somebody has taken some yeast and they've stuck it in a blender with some enzymes and kind of digested it. So whatever is in the yeast cells can be fed to that bacteria. So you're looking at amino acids and nucleotides and salt and a few other things. And there's lots of recipes out there for growing organisms. E. coli likes this terrific broth, and so do a lot of other bacterial organisms. So the reason why we grow bacteria in these undergraduate labs is because not only they're cheap, but they grow really, really quickly. So E. coli will replicate itself in about 30 minutes, even 20 minutes under optimal conditions. So you can imagine after 30 minutes, we have 2 cells. After 60 minutes, we have 4 cells. After 90 minutes, we have 80. Under 30 minutes, we've got 16 cells. And if you keep doubling and doubling and doubling, we can actually grow a culture very easily and very simply just overnight. If we're growing mammalian cells, we'd be looking at 4 to 8 weeks to grow a decent culture, and it would be a lot more laborious and also expensive. Okay, so a couple other things that you're going to see when we start looking at bacteria in the lab. And one thing that we talked about already are these agar plates. So I just want to talk about that. What is agar? Agar is actually a carbohydrate from seaweed. That's what you're looking at there. That's seaweed. So the carbohydrate is extracted and it comes in a powdered form. Sometimes it's a little bit yellowish. It looks a little bit whitish there in that picture. And it's kind of like making Jello. So you take the agar and you put it into a water solution. You also add some broth, so some food in there. It gets boiled, and then it gets poured into the plate, and then you end up with your agar plate. And the agar plate has food in there as well. And so this is kind of a firm surface for growing microorganisms on there. So what are we doing with agar plates? Well, our first lab, so last week we did this environmental survey, and I found that actually Google did something similar to celebrate Julie's Petrie, the inventor of the Petri dish. So I'll play this little video for you. You can see what Google did. So there goes Google. They've got some Petri dishes. And they're spreading something on each Petri dish. But they're doing it in a certain way because this is Google, they're a corporation, so they wanted to make a corporate logo. So there we go, Google. You can see what they swabbed. I'm going to show you here in a second. They swabbed an old sock. Looks like a doorknob, keyboard, a dog's mouth, a plant, and a dish sponge. So I thought that was a cute little video. I thought I would share that with you. So something that you saw in the results from last week's lab are these colonies of bacteria. And the reason why we do it in agar plates is because we can actually make them out, right? If you had this in a test tube and you just have a broth, it's just going to be soupy and turbid, and you're not going to be able to make out that you have several different species. This is a plate here. You can see actually they're making out about 12 different bacterial species from this plate. So I thought that was a nice one to show you. These species come in many different shapes and sizes. So this is a little graphic again from lab 4. So I'm kind of just foreshadowing lab 4 that we're going to get a chance to use some of these words to describe the different types of colonies of the different bacteria. Some of them are very distinct. So if you take a look here on the left is E. coli, and on the right is something called bacillus. They're a very similar color, but you can see that they have a different texture to them. So E. coli is always kind of a little bit shiny, and the shape of the colonies is usually relatively round, whereas bacillus is a bit more flat and not quite so shiny looking. We also talked about the different types of staphylococcus as well. So I found some other different types of bacteria on the internet. I'm not even sure which ones these are, but they're very cool looking. So sometimes you can see these very interesting, unique colonies. Sometimes it's pretty easy to tell what you have. Oh, there's the staphylococcus. So remember Staphylococcus aureus. So there it is, aureus. Remember a U stands for gold. And so the colonies have a bit of a golden color. That's how I know which staph I have, either white or yellow colonies. Or you can make art. There's an entire website devoted to microbial art. So go check it out if that's something you're interested in. So what else is solid media good for? Solid media is good for streaking. So here's a word to the wise, okay? If you're on the internet and you're going to Google Images, always think about what you're typing in Google Images. So I went to Google and you type in streaking, and of course, well, what do you get? You get like the Super Bowl. And I was like, wait a sec, whoa, that's not the kind of streaking that I'm looking for. I'm actually talking about microbial streaking. So put that in, bacteria streaking or microbial streaking, to make sure that you're getting the right search on the internet. So I'll show you what microbial streaking is. Again, we'll do this during lab 4. We're going to do some streaking, and this is going to, the whole idea is to get colonies for isolation. So what do you do here? I'll draw you a little diagram. Here's my little Petri dish. And so what you do is you take some culture and you kind of smear it on one corner of the Petri dish. Then you take your instrument and you sterilize it. And I will demo this in the lab. Then you do another streak and you sterilize it and you do it again. And then maybe one more time if there's enough room on the plate. So what are you doing here? You're kind of, you're diluting your sample each time. As you sterilize the instrument, you're putting it in there and you're smearing it or streaking it across. And the whole idea is so that you get these individual colonies. This way you can make sure that you have you don't have a mixed culture with different species in there, and also that each isolate you have is a genetic clone to itself. And so, like I said, we're going to talk a bit more about this in Lab 4. A couple other things to talk about. So how do we culture animal cells? They tend to be a lot more difficult. Bacterial cells tend to grow in the wild and they will grow free. So you just need to feed them food. Animal cells don't grow like that. Most of our cells grow and they're attached to bone or ligament or some sort of connective tissue. In fact, it's a very good thing they don't just grow in the wild free, because our bodies would be this massive, we'd just be giant tumors. Our cells have very good checks and controls to make sure our bodies are very healthy. So how do we grow animal cells? You have to buy these special plates, and these special plates actually are coated with something like collagen or bone-like substances to allow the cells to grow there. And they'll often just grow as one layer, because that's what animal cells generally do. They also often require complex nutrients because they're used to being in our body, and our body does some really nice things for our cells and provides some good nutrients for them. Specially coated surfaces, I mentioned that. The other misfortune about animal cells is often they only divide a specific number of times. That's what our cells do. They grow a certain number of times, and then that's kind of the end of them. So that makes it a little bit more difficult too. So sometimes we have to figure out how to make our animal cells what's called immortal, to grow them in the lab. And I think I've got a slide on that on the next page here. And some cells don't divide at all. So this is a big kind of issue when people are studying different types of cells, is trying to figure out how to grow them. Sometimes people will spend 5 to 10 years just to figure out how to grow a certain type of animal cell before they can actually study it. So very, very complex. So how do we make cells immortal? We use cancer cells. You may have heard of these particular cells. These ones here are very famous. These are called HeLa cells. You can see HeLa down there. And where did these cells come from? They were isolated from a woman in the 1950s. So you can see her name there, Henrietta H.E. Lacks. So HeLa cells. So this woman had cervical cancer in the 1950s, and her doctor took a biopsy in her cells. We've been growing them ever since. Unfortunately, there's some issues around this story, and usually when doctors take biopsies, the sample gets labeled, and it's not like they go and tell everybody, whose cancer cells they are. You're not allowed to do that anymore. But back in the 50s, these cells were so remarkable, and the name got out, and this coding system is using the person's name in the code isn't actually, you're not really allowed to do that nowadays. Back then, you were able to do it. The woman died. Eventually, reporters went back to the family, and they didn't understand what was going on. They were a very poor Black family, and it was a very interesting human story. If you're interested in these kind of things, I really recommend this book here on the left here, The Immortal Life of Henrietta Lacks. Very fascinating book. I think there was even a movie done on HBO with Oprah in it, if that's something you're interested in. But I highly recommend this book. Very interesting human story, and kind of, a little bit about her cells. This is more about the human side of things, but the cells were very fascinating because they were the first time we could effectively really grow human cells, and it led to all sorts of vaccines and other discoveries starting in the 1950s. So what about culturing other things like viruses? Why do we want to do that? Again, make vaccines, study them. That's a big, important part of viruses. How do we culture viruses? Well, viruses don't eat food. They're not cells. They're not usually considered living organisms. So how do we culture them? We have to feed them cells, basically. So they only grow in cells. So therefore, we have to culture them in cells. So I'll show you some methods for doing this. One method is if you're culturing a virus that infects bacteria, well, you grow up bacteria. And lots of people who have done microbiology have grown up these particular viruses. They're called bacteriophage. So this means bacteria. Phage means eating. So that word really means eaters of bacteria, and it's just a word for viruses that infect bacteria like E. coli. So you can see what we have here is a bacterial lawn, and a lawn is just a whole spread of bacteria across the plate. And those little holes that you see actually represent where the virus is killing the bacteria because the bacteria is not growing. So that's one way to do it, and you can do this with animal viruses as well. You can grow them on those special plates, and you get these little holes, and the holes are called plaques, by the way. You can stick a syringe into that hole, and in that liquid, it will be full of viruses. Here's the term plaque. You may have heard of some animal viruses being cultured in eggs. So that's what this is showing here. And actually, there's a number of human vaccines that are cultured in what are called embryonated chicken eggs. And so if you go for your flu shot, they will usually ask you, hey, are you allergic to eggs? Because they want to know, because they don't want to be giving you something you might be allergic to that could be in the actual vaccine. So this is a very useful technique. We also use HeLa cells. Again, it just depends on the type of virus and the vaccine, and sometimes that's part of the research, is trying to figure out how to culture virus. Some viruses we can't culture, we don't know how to do it. You know, other than infecting humans, which may not necessarily be that ethical. This is just saying, hey, you can grow them in those cancerous cells. Okay, one other thing that I'll probably discuss in lab and lab 4 is something called aseptic technique. And what does that mean? It means we're trying to be sterile when we're handling things in the lab. And why would we do that? Well, two things. We're trying to protect us. Okay, if you're in my lab and you're dealing with bacteria that are potentially infectious, I want to make sure that you don't bring it home, get it up your nose, in your eyes, in your lunch, those kind of things. And the other thing I want to be sure is that you are also not infecting your experiment. We have bacteria all over our skin. We're breathing out bacteria out of our mouths. So you want to make sure you are protecting your experiments. So I'm going to be talking about this a little bit when I'm manipulating the bacteria in the lab for lab 4. So something else to look forward to. And if you take any microbiology classes or other cell biology classes, you're going to learn some of these techniques. You can see in this diagram here, some of those techniques include wearing gloves, maybe a face mask or a beard mask. Gentlemen, if you do have a beard, there's a lot of skin cells that do hang out in there, and sometimes people with beards have issues contaminating their samples, so they might wear masks to prevent that. So one last thing to talk about in this unit is, how do we study these things? How do we break them apart? This is kind of, you know, one last component to this. Again, something that we really can't do in an undergraduate lab because this usually takes like 5 to 16 hours. You can't do it in a 3-hour period. But it's an important technique to know a little bit about. It's called cell fractionation. So how do we get those organelles so that we can study them one at a time? First, you break them apart. How do we break them apart? Well, there's a whole bunch of different ways. There's mechanically, we can use things called, we can use blenders, basically a kitchen blender. Sometimes people use something called sonication that uses high frequency sound. We can use detergents, and eventually we get some sort of mixture of those organelles and the other cell components in the test tube. So the next step of the cell fractionation is to basically use a centrifuge to spin out the components of the cell. So there's many different types of centrifuges. You've probably seen some of the low-speed ones used in high schools. You can buy these really, really fast ones. You can see this one here is called a swinging bucket, but the tubes actually spread out 180 degrees from one another. And then basically you spin at different speeds. So we can get out nuclei relatively easily, and from some cells from relatively low spins. You could do this with a high school centrifuge potentially. You can see 1,000 times G at 10 minutes. If we want smaller organelles like mitochondria, you're already getting into specialized centrifuges, 20,000 G. So that's about 20,000 revolutions per minute, which is very, very fast spinning. Smaller things, you're going to go even faster. You can see 80,000 G for microsomes and 150,000 G for ribosomes. So I used to do a little bit of this back in the day, and the protocols were all 16 to 24 hours. They were just ridiculously long experiments and very painful. But if you get good results, then that's great. Okay, so that's it for topic, what is this, topic 3. And so slightly behind where I wanted to be, but that's okay. I'm going to switch now to topic 4. And topic 4, we're going to talk about membranes, which, just make sure, okay, how do I, just need to share my screen here. So topic 4 is kind of about 3 lectures, sometimes 2. Will probably take us 3 lectures to get through it because we're starting late today. We're going to talk about cell membranes, we're going to talk about membrane transport, and we're going to talk about other cell surfaces. And so your lab 3, which is next week, is going to be on cell membranes. And it's our timing is really good on this. And we're going to talk about more about bacteria and the bacterial cell wall, and that's going to be lab 4. So a lot of this stuff will tie into the lab, which is a good thing. So let's talk about membranes. See here, there we go. So what is a membrane? You probably know this definition of a membrane, right? It's a selectively permeable lipid bilayer that separates a cell from its surrounding. So we're going to kind of delve into what all that stuff means, right? You know, lipid bilayer, and you can see from this diagram that it's not just lipids. In fact, about 50% of the mass of a typical membrane is protein. So it has a lot more to do than just these lipids. Notice another term that I'm giving you here, plasma membrane. We're going to use that term a lot more than cell membrane because there are many cell membranes. We've got membranes that surround all these organelles. We have membranes that surround the nucleus, and all those are technically cell membranes. So the plasma membrane is the outermost one. So just to distinguish between those 2 words. And we will talk about membranes a lot more in topic 6 when we talk about organelles. So something to consider is what membranes are made of. They're made of proteins, they're made of phospholipids, and they're made of carbohydrates sometimes. So we're going to talk about all 3 of those components. This diagram here has a couple of other things. The cytoskeleton on the inside, that's topic 7. And the extracellular matrix, we're going to cover that when we get to cell walls. It's something that's outside of the cell membrane. So we're not really concerned about the extracellular matrix today, but we will get to it. Okay, so let's talk about that first component of the cell membrane, those phospholipids. We've talked about phospholipids already. You can see there's the structure. So just a reminder of what the structure of a phospholipid is. If you take a look at this, we've got this little thing here in the middle is glycerol. And I have some fatty acids. So there's 2 fatty acids, 1 and 2. You can see that second fatty acid that has a double bond, it's unsaturated. And we don't have the third fatty acid, so this is not a triacylglycerol. Instead, we have this phosphate group. In fact, this here is phosphatidylcholine, and there's different phosphate groups. There's phosphatidylserine, and there's a whole bunch of them anyway. So look for that phosphate. That's always a clue that you have a phospholipid. So this whole thing here kind of looks like it's got a head. You can see there's my head. And it's polar, it's got some charges, negative and positive charges, and then it has these 2 legs. And so over here, you can see how they're trying to represent the phospholipid is this polar hydrophilic head with the 2 hydrophobic tails. So I want to introduce a new word for you. You can see the word is up here, amphipathic. So usually I write a definition on the board for amphipathic. But I'll just tell you orally what that means. It means a molecule just like a phospholipid. It's a molecule that contains hydrophobic and hydrophilic regions. So there are a few types of molecules like this. Detergents fall into those categories. And so they kind of have dual properties, right? They have a polar area that can interact with water, and they have the hydrophobic area that can interact with hydrophobic substances. So this is important for membranes because it's kind of in their nature how they assemble together. So you stick these phospholipids in water and they form these bilayers. And so you can see the bilayer has this polar zone where the heads are, and then in the middle it's got this hydrophobic zone where all the tails are. And so it kind of has this dual nature, and it's a great way to separate the cell from its surroundings. So we're going to talk about archaea a little bit in the next unit. We mentioned them a little bit at the beginning of the course, is that they're kind of like bacteria, but not quite. So one way that they are different from bacteria and archaea are their phospholipids. So if you take a look here on the left, I'm showing you what a phospholipid bilayer looks like in bacteria and eukaryotes. You have these fatty acids. The fatty acids are unbranched, and they're attached to the polar heads in something called an ester linkage. If you look at archaea, some archaea have really weird phospholipids. First of all, they're branched. So that means the carbon structures on the fatty acid tails have these branches on them. And secondly, some of them actually form these weird monolayers. So this is very different. These organisms biochemically are something that are unique from the bacteria and the archaea. Why do some of them have these monolayers? Some of them are these thermophiles. They live in these extreme environments. And so if you don't want your membrane to melt, you're doing something different to make sure that it's not damaged as much by heat. And so that's why they have these. They also have ether linkages. If you don't know the difference between ester and ethers, don't worry about that in this class. Worry about it when you take organic chem, because you're going to talk about organic chemistry. Okay, so one unique thing about the archaea. More on them in topic 4. So the second component are proteins. So if you're looking at the percentage, like I said, it's about 50% phospholipids by mass, and membranes are about 50% proteins by mass. About. Kind of depends on the type of membrane. If you take a look, we have these proteins embedded, and these embedded proteins have hydrophobic regions. So if you remember when we were talking about amino acids, we have all these amino acids, and some of them are polar, some of them are non-polar. So all the amino acids are going to be found in this embedded region are going to be non-polar amino acids. The ones on the ends, this is where you're going to find the polar amino acids. And so it's kind of like phospholipids. You can have amphipathic proteins that have polar and non-polar components. Some terminology for you. A lot of proteins are embedded, and we call those integral membrane proteins. So integral is kind of like integrated, meaning they're integrated into the membrane. Many membrane proteins fall into this category. Some of these proteins are kind of loosely associated with the membrane. So if I did a cell fractionation and I made a membrane preparation, I would get these peripheral proteins that come with the membranes, but they're not embedded. They're peripherals, which means they're sort of attachments, but they are important membrane proteins. So just some terminology for you. Sometimes we use the word transmembrane. You can see there's the word transmembrane. And a lot of these transmembrane proteins, the structure that is going through the membrane is actually an alpha helix. And this alpha helix here, by the way, this represents about 20 amino acids. And so that's something you can see just looking at the genetic sequence. If you see a string of 20 amino acids that are all hydrophobic, usually it's assumed that you do have some sort of membrane protein, of which a lot of proteins are membrane proteins. There's a bunch of different systems for these membrane proteins. Some of them have 1 alpha helix across the membrane, some of them have 3, some of them have 7, a bunch of different ways to do this. And just thought I'd show you this picture representing some different membrane proteins. Here's one protein that I used to work on. This is called a penicillin binding protein, and most of the protein is not associated with the membrane, but it has 1 alpha helix, which is kind of like an anchor. So I never really called this a transmembrane protein. We used the term anchored protein. So just something a little bit different. So what are these membrane proteins doing? A whole bunch of things. If you think about that list of things that proteins are doing in general, well, many proteins are membrane proteins, so they're just doing the same functions. Some of them are transporters. We're going to talk about that next day, these transport proteins, and they're bringing things inside and outside of the cell. Many membrane proteins are enzymes. They're just attached to a membrane, but they're an enzyme. Many are receptors for things like hormones. So hormone is a molecule that might travel through your blood from one tissue to another. And these receptors are receiving the signal, and they're transmitting the signal inside the cell. So it could be insulin, which is telling your muscle cells time to take up glucose. Some of them are involved in cell-to-cell recognition, so the immune system is recognizing different cells. Some of them are like joints, so they're like rivets holding your tissues together. And some of them are attaching to the, I guess that's very similar, attachment to the cytoskeleton extracellular matrix. They're attachment proteins. So membrane proteins, many, many different tasks. Okay, so the third component. So I told you we're looking at about 50-50, right? Phospholipids and 50% proteins. So how do carbohydrates fit in? Well, they're small, so maybe 1 to 2%. And they're not even found in all membranes. There's different types of membranes. Some of them are what we call glycoproteins. So what is a glycoprotein? Well, it's a protein that has a glyco component. Glyco, of course, is a carbohydrate. So you can see this image here. We've got that embedded protein, and that little green structure stretching off it is a carbohydrate. Some of them are glycolipids. So same idea. Now you have a phospholipid, and there's some carbohydrate attached to it. And these things are made through a process called glycosylation. So there's that word glyco, which means carbohydrate. Glycosylation means the process of adding a carbohydrate. So what are these carbohydrates doing? Notice the first thing is that these carbohydrates are only on the outside of the cell. And I know I mentioned it last week at some point. I mentioned that carbohydrates are kind of, they're kind of like tags. You can kind of think of them as luggage tags. The little tags that say, you know, this is a skin cell, or this is a blood cell, or this is a spleen cell. And this is really important for multicellular organisms. You know, our body differentiates into different cells and different tissues, different organs, and it's really important for our body to, you know, have these labels so that we can make sure we grow our bones the right size and our heart in the right shape and those kind of things. It's also important, though, in medicine because, of course, you know, we're looking at compatibility of tissues. If people are getting some sort of blood transfusion or some sort of transplant, we've got to make sure they're compatible, because if they're not compatible, you're going to have a very severe immune reaction. So that would not be good. Okay, so I don't have a Kahoot for you today, but I have a couple of these test yourself questions. And I wanted to show you these kind of questions because we're starting to think about the midterm, which is about 2.5 weeks away, I think, if I remember correctly. And it's important that you think about how to answer multiple choice questions. Take a look. Here's a multiple choice question, and it may look difficult or it may not look difficult to you. I personally don't think it's difficult, but then of course I'm familiar with the question. I wrote it. But you do have to read these things carefully, and you have to make sure that you understand all the language in the question. So let's go through these one by one and make sure that we understand this question. So it says here, which of the following is not true? So which one is false about membrane proteins? Okay, so let's take a look. So the first question, integral proteins are found embedded within the phospholipid bilayer. So remember, integral means integrated or embedded. So this one is good. So we are happy with that one there. Question 2, peripheral proteins. So which ones are those? Those are the ones that are loosely associated. So by definition, this one is correct as well. Okay, that one's good. C, membrane proteins can be channels that allow for molecules to pass the phospholipid bilayer. That's also true. Many membrane proteins are in fact channels or transporters. So that one is good. D, integral proteins must be comprised of mostly hydrophilic amino acids. Okay, so which ones are the integral proteins? They're the ones that are embedded, the ones that are found in the middle of the phospholipid bilayer. And remember that our phospholipid bilayer is made of mostly hydrophobic amino acids. So this is the wrong word. So that is the one that is not true. Let's just take a look at E. So don't always stop if you think you found the right answer. Make sure you do read all the options. Peripheral proteins can be enzymes. So peripheral proteins are the ones that are loosely associated. Can they be enzymes? Of course they can. Many proteins in the cell are enzymes. So that one is good. So do make sure you read these questions. And like I said before, it's very important that you do understand all the terminology so that you can read and interpret the questions. So make sure you do look at your vocabulary at the end of every unit to make sure that you do understand all the words. Because I'm not going to necessarily use simple words. I'm going to use the words of the course when I write the questions. So do make sure that you understand the words of the course. Okay, so where are we at? 22. So I'm going to spend the next 10 minutes kind of talking a little bit about the fluid mosaic model. This is kind of the current model of a cellular membrane. This was, you can see, was proposed back in 1972. There are previous models of the membrane that kind of had other things going on. And this is sort of what we've been showing, is this whole idea that we have these this phospholipid bilayer, and we have these embedded proteins. So take a look at the word fluid. What does that mean? It means liquidy. I want to talk about that in a moment. And the liquidy part has to do with the phospholipids. The mosaic has to do with these embedded proteins. A mosaic is kind of like a patchwork thing. So you can think of it as like a quilt of proteins. So this fluid mosaic model, by the way, was confirmed. One of the ways they confirmed this was doing a special kind of scanning electron microscopy technique called freeze fracture. It's actually a really cool technique where they freeze a sample and then they basically peel it apart, kind of like a banana. So what's going on is that the cells actually break apart at the area of least resistance, and that area of least resistance is right in the middle of the phospholipid bilayer. So they peel them apart, and they'd be able to observe all these embedded membrane proteins. So you can see there's a couple of pictures there showing these membrane proteins that were very, very visible. Another experiment that also confirmed the fluid mosaic model, the fluid part of this, were these fusion experiments. So what they did is they took two cell types. I don't know why they used a mouse and a human cell. It doesn't really matter. And they stained them with fluorescent dyes. So the mouse cell was stained one color. The human cell was stained another color. It actually wasn't pink and purple like they're showing in this diagram. It was actually green and red. I think they're getting away from green and red in textbooks because of people that are colorblind. But anyway, it doesn't really matter. Two different colors. You can put them in a petri dish. You add a little bit of detergent, and the cell membranes actually kind of fuse together. And they can watch this over about an hour. What happens are the colored proteins actually start to mix and move. So they observe that the cells were in fact, or the membranes in fact, were kind of fluid. I have a little animation here. I'll show you the animation. It's pretty low tech, but you can see the whole process happening. Those proteins, those embedded proteins, are moving around like little icebergs in an ocean. Right, an ocean of phospholipids. So let's talk about this fluid stuff here, okay? I keep mentioning that phospholipids are fluid. So they're fluid in that kind of like a picture, like a thick salad dressing, right? That's what a membrane is like, meaning that those phospholipids are together and they can move around mostly laterally. Bilaterally, I mean kind of back and forth and side to side. So you can see that it's showing on this diagram here, about 1 million, 10 million times a second, they're moving and changing place, kind of like water flowing and moving back and forth. They don't flip-flop though. Flip-flopping meaning going from one side of the bilayer to the other, because of course that would involve bringing a polar molecule through a hydrophobic zone, and that doesn't really happen. I'll show you, I've got a little video here. Here it goes. And you can see our little phospholipids are moving back and forth and then moving laterally. You can also see the embedded proteins are moving back and forth laterally. They can also twist a little bit too, right? It's not really hard to show in that video there, but they can actually twist back and forth as well. So that's the fluid part of the fluid mosaic model. So a couple other things to sort of finish off today is what is determining fluidity? A couple of things that are determining fluidity are the type, size and shape of your fatty acids. That's what FA stands for, is fatty acid chains. So generally, the longer the chains, the less fluid. So what's going on? Longer chains, you've got larger molecules, you've got more van der Waals and London dispersion force, so you've got more solid fatty acids. So if you want more liquidy, you're going to make shorter fatty acids. So it's going to depend on what environment you're living in. Are you living in a cold area or a warm area? Another thing that's important is the saturation of the fatty acid chains. So we talked a little bit about this already. Saturated means all full of hydrogens. Unsaturated means they have double bonds. You take a look, here's another picture showing the unsaturated on the left, and those double bonds add these bends or kinks. And if they're bent and kinked, they're not packing very well. In fact, they're sliding around a bit more and then more liquidy or more fluid. Another thing that determines membrane fluidity is the presence of cholesterols. It's a little complicated what cholesterols are doing, but if you take a look at cholesterol, notice it's mostly hydrophobic. It's got that little OH on the end, so it's got a very small polar section to this molecule. This is a steroid, by the way. Remember, we were talking about steroids, and you can see it fits right there into the membrane. So what is cholesterol doing? It's kind of plugging holes, is what it's doing. So if the membrane gets a little too fluid, it plugs those holes and helps to kind of stabilize the membrane. If the membrane gets a little bit too hard, it's actually helping keep things loose. So it's doing a little bit of both. It's kind of this homeostasis mechanism, so that your membrane, you want a certain amount of fluidity, but not too solid, not too liquid. And what cholesterol does is it sort of broadens that out, so you can be in a state of homeostasis at a broader temperature range. You can read about it in the textbook. I'm not going to go into it in really any amount of detail other than that. But know that cholesterol is important for membrane fluidity. Okay, let me just see where we are here. Almost done for today. I wanted to show you something else that can affect membrane fluidity are chemicals. So you may have heard of a little chemical called alcohol. Alcohol is, of course, people will drink it, and we have an enzyme called alcohol dehydrogenase that usually breaks it down. But if you, of course, consume lots of alcohol, or if you consume it really quickly, you're going to have physiological side effects. And so what is that alcohol doing? Well, it turns out alcohol can get into the phospholipid bilayer. And so you may know that people who are under the influence of alcohol, when they're driving, their reaction times are slower. That's because the ethanol is actually getting into the nerves and slowing down the nerve transmission. If people even go further beyond that, this can actually be detrimental to their body and can cause damage to the liver, and alcohol intoxication can be a very serious thing. So just wanted to show you that. That's not on the exam, but just something that does affect membrane fluidity. Okay, I'm just going to finish off kind of saying, I have had this kind of midterm question in the past. I'm probably online. I'm not going to be asking you to draw something. I know in the past I've had this kind of question. I can't remember the exact wording, but it said, with the aid of a diagram, describe the fluid mosaic model, and maybe I asked you something else. I really can't remember. So keep this in mind, when I'm talking about something like the fluid mosaic model, what this should include. We were just talking about a whole bunch of things, cholesterol and glycolipids, and those things are not as essential to the actual fluid mosaic model. Definitely you want to include phospholipids, right? So if you're going to do a diagram, you can draw these little things, right? So here's my little diagram. I can add some sort of protein. There's some more phospholipids. So this isn't good enough, right? You can't just draw a diagram and leave it and just assume I know that you know everything. Your job is to convince me. So label this, right? So you can label this, and you can say this is the polar phospholipid head, right? We've got non-polar fatty acids. I'm going to label this as a integral membrane protein. Maybe I'll label this as a hydrophobic zone. And then there's going to be, you know, some sort of description on the side, right? So I'm going to have my diagram. I'm just going to label it, and then I'm going to talk about, you know, what's going on with the, you know, the hydrophobic zone and the polar zone, and maybe mention what's going on with amino acids and those kind of things. Okay, I see our time is up. I see there's one question here. Somebody asked if there is a pre-lab for tomorrow. No, there is not. I have tomorrow's lab ready. I'll be sending out around noon tomorrow, and there's no pre-lab for it. So this basically, it will pick up from last day's lab with some new questions, and then there's an assignment to hand in that's found at the end of the lab. So once that comes out, you can take a look at that and see what's involved. Okay, so I guess that's it for today. I will talk to some of you tomorrow. I will have an office hour as well, like I had last day, with for the lab for any questions. Somebody's saying, do we need to hand in lab exercise 1 with tomorrow's lab? Sort of, right? So tomorrow's lab, the actual assignment is at the end. So make sure you take a look at that. It has questions that are from lab 1 and questions that are from lab 2. And those are going to be combined as one assignment. And it is due, and that will be due 1 week from tomorrow, which is next Thursday. So I think that's the 24th of September. Okay, so if you have any more questions with the lab, do join in my office hour tomorrow. I will send out the link. It's the same one I sent out with last week's lab. I'll be available by Zoom to answer any questions. Other than that, I hope you have a great evening, and I will see you then.